This is a biochemistry lecture on steroid biosynthesis. My name is Jeff, and I'm a professor at Kansas City University. For our first learning objective, I'd like to point out that the reading reference for you today is an illustrated review of Medical Biochemistry by Panini. For learning objective 1, we need to describe the general aspects of steroid hormones. Listed here are certain pages and figures you can read. Page 36, figure 3.8, pages 247 through 248, figures 14.13 and table 14.3. There are 3 correlation boxes on page 247, 2 orange and 1 blue. In particular, the blue one describes infant respiratory distress syndrome and the use of glucocorticoids in this clinical condition. Now I'd like to turn you on to something called the Kyoto Encyclopedia of Genes and Genomes, or KEG for short. Here's a URL which you can click on, and if you typed in steroid biosynthesis, an interesting table would come up, and I'll show you that in the next slide. What this is a collection of online searchable databases dealing with genomes, biological pathways, diseases, drugs, and chemical substances. So when you type in steroid biosynthesis, this is what pops up. And here's that link for your own personal interest. And all of these enzymes are clickable. What we're going to focus on is this little corner. You can see cholesterol is quite the center point. So let's blow this up a little bigger. When you do that, cholesterol can be converted into steroid hormones. There are pathways of steroid degradation, primary bile acid synthesis, vitamin D synthesis. Vitamin D goes from our skin through an intermediate called 7-dehydrocholesterol through the actions of light. So what I'd like you to know now is all steroid hormones. So this slide provides you with a pictorial version of what I said in the previous slide with a little more detail added. First of all, the rate limiting enzyme is here. It's called desmolase. Again, it converts a C27 or a 27 carbon molecule into a 21 carbon molecule. You'll note this side chain up here is very different. So here are the synonyms of desmolase. 2022 desmolase, also called cytochrome P450 SCC, which stands for side chain cleavage, also known as CYP11A1. This enzyme is found in all steroid-producing tissues, including adrenal glands, testes, ovary, and placenta. It catalyzes the first and rate-limiting step in the conversion of cholesterol to steroid hormones through an intermediate called pregnenolone. It's regulated by ACTH. Okay, students, you do not need to know this. Okay, so pregnenolone becomes a very important biochemical intermediate. It's used as the substrate to produce all other steroid hormones, including progesterone, aldosterone, cortisol, testosterone. Okay, now you should have a foundational understanding of the biochemistry. Okay, AAFP, both classic and non-classic forms of this disease. Both forms of adrenal hyperplasia we're going to talk about are caused by deficiencies in the adrenal enzymes that are used to synthesize glucocorticoids. The net result is increased production. Okay, folks, I pulled this from Panini, your textbook. We're focused in on the adrenal gland. There's a cortex and a medulla, zona glomerulosa, fasciculata, reticularis, and you'll see those words here, glomerulosa, fasciculata, and reticularis. That's the areas of the adrenal gland where these enzymes are focused and these conditions are manifested. Those things listed in italics are the enzymes. Specifically, we're going to talk about 21 alpha hydroxylase and 11 beta hydroxylase with reference to the renin-angiotensin system and the effects of aldosterone and some of these other intermediate steroid hormones that they travel through. For example, cholesterol goes to progesterone, goes to corticosterone, eventually to aldosterone. When you have mutations in these genes, certain physiological outcomes result. And so I'm going to focus you now on what you need to drill into. For example, it would be good if you knew that desmolase is regulated by ACTH and the function of 1720 lyase, 21 alpha hydroxylase, and 11 beta hydroxylase, and their role in producing congenital adrenal hyperplasia, severe forms and more mild forms of the disease. Moreover, I show you here the structure of the hormones that we're talking about. Women present with hirsutism. Hirsutism is excessive body hair in both men and women, where it's normally absent. Female patients also present with general. So at this point, you may be asking yourself, how do we diagnose these patients? More importantly, how do we determine if a developing fetus is at risk for congenital adrenal hyperplasia? Well, the first way is to do some HLA haplotype. We determine that looking at human leukocyte antigen and determining what type it is, and that can tell us the extent to which the developing fetus is likely to have congenital adrenal hyperplasia. The second treatment of these patients includes carefully monitored hormone replacement therapy. That demands a prompt recognition of the problem to get the timely replacement therapy on board, and has Now let's go deeper into the biochemistry of congenital adrenal hyperplasia. What I'm showing you here is an alternative view. Here's desmolase, 17 alpha hydroxylase, C17 lyase. Cholesterol is converted eventually to dihydroepiandrosterone through pregnenolone and 17-hydroxypregnenolone. These compounds are converted by various enzymes to aldosterone, cortisol, and testosterone. You see, these are all branch points. Many of these enzymes listed here are cytochrome P450s, including 21-hydroxylase, and mutations in this gene have a profound effect on congenital adrenal hyperplasia, as well as let's look a little bit deeper at 21-hydroxylase deficiency. 21-hydroxylase is also known as CYP21A2, and when it's deficient, in other words, when this enzyme is mutated, there's a blockage. Thus, progesterone and 17-hydroxyprogesterone accumulate. In other words, when this enzyme is dead, there is no longer conversion of progesterone to aldosterone, nor is there conversion of 17-hydroxyprogesterone to cortisol. Similarly, when the 11-beta hydroxylase is mutated, there's a blockage at this level. That means there's an accumulation of deoxycorticosterone and about aldosterone's hormonal effects. This hormone is produced in a tissue-specific manner from progesterone. Progesterone is normally converted to aldosterone in a wild-type situation, that's without a mutation, and this hormone aldosterone binds to and activates the mineralocorticoid receptor, which is a very important nuclear receptor superfamily member. In the next slide, I'll tell you about the molecular mechanism of nuclear. This slide depicts how nuclear receptor superfamily signaling works. Small lipophilic greasy hormones bind to a red inactive receptor. The complex shifts conformation, in other words, the circle turns to a square, which binds to DNA to regulate nuclear receptor target genes. Now let's talk about primary aldosteronism. It's a defect in the zona glomerulosa of the adrenal cortex. High levels of aldosterone promote retention of sodium and water in the kidneys, and moreover promotes excretion of potassium ions. Aldosterone in excess levels, such as occurs during primary aldosteronism, also called Conn syndrome, increases blood volume and thus produces hypertension. Again, Conn syndrome. It's an endocrine disorder of the adrenal glands in which there's an excessive secretion of the hormone aldosterone. That leads to a retention of sodium, a loss of potassium. Now I'd like to talk about 11-beta hydroxylase deficiency. That's a condition in which this enzyme at this step is knocked out. What you see in those patients is a rise in the levels of 11-deoxycorticosterone and 11. So it turns out many steroids can activate mineralocorticoid receptor with variable efficacy. Aldosterone has the highest, followed by 11-deoxycorticosterone, and testosterone, hydrocortisone, and cortisone have their own efficacy. All of these hormones. So the paradigm becomes increased aldosterone signaling leads to hypertension. But in 11-beta hydroxylase deficiency, there's kind of this paradox. They have decreased aldosterone synthesis with hypertension. How is this possible? Moreover, their hydrocortisone and cortisone synthesis decreases because the synthesis of those hormones is dependent on 11-beta hydroxylase activity. So that's further adding to the conundrum. So it turns out 11-deoxycorticosterone and 11-deoxycortisol levels increase. And it turns out 11-deoxycorticosterone has a relatively high affinity and efficacy for mineralocorticoid receptor. So what I give you here is a summary of congenital adrenal hyperplasia. Mutations can occur in all of these genes, including 3-beta hydroxysteroid dehydrogenase, 17-alpha hydroxylase, 21-alpha hydroxylase, and 11-beta hydroxylase. This is a pretty good chart that emphasizes a lot of differences and commonalities among these different mutations, all of which cause the syndrome, congenital adrenal hyperplasia. I'd like to highlight this one. We're now moving on to learn. Now let's go a little bit deeper and get a better understanding of ACTH and its role in regulating cortisol production. We're going to use ACTH for purposes of the exam, but it's also known as corticotropin or adrenal. Now I'd like to give you a little more of the biology of ACTH. First of all, it's a 39 amino acid peptide secreted by the anterior pituitary gland. ACTH stimulates the production of cortisol, and that cortisol is important not only for maintaining blood pressure, as we've talked about, but also stimulates regulation of glucose, protein, and lipid metabolism. Moreover, cortisol suppresses the immune system response and helps maintain blood pressure, as we've already talked about. Additional information about ACTH can be found here. The adrenal glands, primarily the adrenal cortex, is stimulated by ACTH, specifically in the region of the zona fasciculata. Again, this stimulates secretion of glucocorticoid steroid hormones, such as cortisol. Now let's look inside cells and see what's happening with the signaling pathways. In the absence of ACTH, protein kinase A is inactive, there's no cell proliferation and no cortisol production. ACTH appears, binds this 7-transmembrane G protein coupled receptor that stimulates the release of G alpha S proteins, and adenylyl cyclase then is free to produce cyclic AMP, which activates the secondary messenger, protein kinase A. Protein kinase A phosphorylates CREB. CREB is a transcription factor that drives cortisol production. Active pKa also drives cell proliferation. What's interesting about all of this is that in certain tumor cells, the regulation of pKa activity is uncoupled from ACTH. So you have constitutively active pKa, which leads to hypercell proliferation and massive cortisol production. So these tumors produce major amounts of cortisol, and the cells proliferate and the tumor progresses. So all of this happens very rapidly, within minutes. Moreover, there's a stimulation of cholesterol delivery to the mitochondria where desmolase is located. And just to recall, desmolase is that So to summarize ACTH function, ACTH I'd like to focus in on steroid hormone synthesis in the adrenal cortex. Again, here's the picture from your Panini textbook, and 1720 lyase is here and here. And we have a couple of things to point out in this slide. CYP17A1 is the same thing as 1720 lyase. In particular, the gene is called CYP17A1 that encodes the enzyme 1720 lyase. And that enzyme has both 17-alpha hydroxylase as well as 1720 lyase activities. It's a key enzyme in the steroidogenic pathway to produce progestins and other steroid hormones. And there are mutations in this gene. They're associated with pseudohermaphroditism and adrenal hyperplasia. Moreover, deficient production of androgen produces 46. I'm showing you this slide just to emphasize that the precursor for androgen production is 17-hydroxyprogesterone and hydroxypregnenolone. You can see that here. CYP17A1 is here, and here are 17-alpha hydroxyprogesterone and 17-alpha hydroxypregnenolone. So mutations in this gene, you'd expect an accumulation of these precursors and a lack of downstream. Now let's move on to our. Returning to the original figure I showed you at the beginning of this lecture, we're going to focus in on 7-D-hydroxycholesterol and the role of light in the conversion to vitamin D3. So we're over on this side of the chart. Now I'd like to talk about the role of various tissues in vitamin D production. Cholecalciferol, also known as vitamin D3, is obtained in two different ways. First, through the diet. Our intestine is able to absorb vitamin D2 from food that we eat, and when it's in the form of vitamin D2, it's called ergocalciferol. Or 7-dehydrocholesterol a storage form of vitamin D in the skin, and it's converted to vitamin D through the action of UV radiation. In the kidney, 1,25-dihydroxycholecalciferol, also known as calcitriol, and that's the active form of vitamin D, is synthesized by an enzyme called 1-alpha hydroxylase. Continuing on with the role of tissues in vitamin D production, whether it's from the skin or the intestine, vitamin D3 travels to the liver, where an enzyme called 25-hydroxylase, which is a liver-specific enzyme, converts vitamin D3 to 25-hydroxycholecalciferol. Thus, 25-hydroxycholecalciferol is found exclusively in the liver. From the liver, it travels to the kidney, where it's converted to 1,25-dihydroxycholecalciferol, which is the active form of vitamin D, also known as calcitriol. That's through the action of an enzyme called 1-alpha hydroxylase, and that enzyme's regulated. Finally, to summarize the role of tissues in vitamin D production, whether you get it from your diet or from your skin and sun, vitamin D is inert when it's in the form of vitamin D2 or vitamin D3. Vitamin D3 is converted in the liver to 25-hydroxy vitamin D. From the liver, it travels to the kidney, where under the direction of circulating parathyroid hormone, it's converted to its active form, which is called 1,25-dihydroxy vitamin D, also known as calcitriol. Okay, that finishes up this lecture. What I'd like to do now is just say thank you